Hey future doctors, welcome to Dentistionary. Here you will be provided with information for the doctors from the doctors. Sorry for this much delay for the salivary glands video, and we are presenting you the salivary glands video before the oral mucous membrane because someone just requested about the deglutination, the phases of deglutition or the phases of stages of swallowing. So you have to study salivary glands first, then the deglutition, and then we'll study about the oral mucous membrane. So let's start with the general introduction. The salivary glands are compound exocrine glands located within and around the oral cavity. These have epithelial secretory units called acini and ducts lined by epithelium that serve to modify and conduct the secretions into the oral cavity. We will deeply learn about the secretory units, acini, and the ducts. Okay, these parenchymal structures are supported by connective tissue that encapsulates the gland and also divides it into the small lobes or lobules. See, these are some lobules. This is major salivary glands. These are the cross sections. Okay, these are the cross sections. So, schematic diagram of a typical salivary gland. The A1 is salivary serous acini. This is the serous acini. This is the cross section. Then B, the serous daminules. Okay, here are the B, the daminules. Then comes the mucous acini, the C1, and the C is here. This one, daminules are above the mucous salivary glands, mucous acini. And then the intercollected duct, the D, intercollected duct, striated duct, and the terminal excretory duct or the main duct, this one. Okay, this is the cross sections. So we'll be studying in the detail about all of these things. First of all, the saliva composition. Saliva is mainly consists of water. So 90%, 99% of water is present in the saliva composition, and the rest 1% is the organic and inorganic substances. The inorganic ions, which are secretory proteins, mainly electrolytes like sodium, potassium, calcium, chlorine, etc. etc. Then comes the organic substances: amylase, ribonuclease, kallikrein, esterase, nystatin, and all these proteins and all. The other organic substances are clotting factors, amino acids, urea, glucose, lipids, and hormones. The saliva also consists of the immunoglobulins IgA, IgG, and IgM for the protection of our mouth from the, because it is the first layer which connects the inside and the outside or the internal environment of the body to the external environment. So the immunoglobulins are very necessary to protect our cells from outside microorganisms. Okay. Also, the mixed saliva or the whole saliva contains secreted saliva and desquamated oral epithelial cells, microorganisms and their products, leukocytes, food debris, and the gingival fluid. This all mixed saliva or whole saliva, this comes because the microorganisms which are killed by these things, okay, the food debris which is accumulated in around the tooth and the interdental spaces or the embrasures you have studied, the gingival fluid released from the gingival epithelium, the leukocytes to protection products, oral epithelial cells that are degenerated. Note that point that saliva produced by major salivary glands differ from one another in a composition. The parotid gland secretes a watery saliva rich in enzymes such as amylase, proline-rich proteins, and glycoproteins. Okay. And submandibular gland contains higher proportion of glycosylated substances such as mucin. Okay, that bit, mucin. In addition to the compositions or the components already listed. The sublingual gland produces a viscous saliva rich in mucin, mostly rich in mucin. Okay, let's follow up. The functions of the saliva: protection and lubrication, antimicrobial action, mucosal integrity, dilution and cleansing, buffer capacity and remyelination, deglutition, digestion, taste, phonation, and excretion. So here in these components, mainly water is the component. The water, the water, 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 water. Everywhere is the water. Then comes for the protection and the lubrication, mucin, protein-rich proteins and water. Antimicrobial actions such as lysozyme, lactoferrin, lactoperoxidases, mucins, cystatin, histatins, IgM, PRPs, IgAs. These all help in the antimicrobial action, or mainly you can say the protection. Mucosal integrity or the mucosal integrity also comes under the protection because if our mouth dries, degeneration of the cells will occur more frequently. So it will harm the mucosa. That's why the mucosal integrity is must. Then the dilution and cleansing, that's by the water. It helps in digestion. Digestion how? The saliva participates in the digestion by providing a fluid environment for solubilization of food, or we call it dilution. And taste substances for the taste, because it increases the surface area and it spreads the taste molecules of the food in the in our papillas, okay, fusiform, filiform, and all. We will study about that in the oral mucous membrane, okay. Then it also helps the action of digestive enzymes, principally amylase, that is useful in two things. That's to digest the proteins, okay, and also ingested carbohydrates to produce glucose or maltose somewhat in 30 minutes. And that's how it keeps our mouth clean with the buffer action because of the antimicrobial action and the lubrication. So it helps in that. It helps in mastication and deglutition. It means saliva moistens the food and helps it break down into smaller particles to initiate digestion. The moistening and lubrication properties of saliva allowing the formation of bolus and facilitate deglutition. Okay, the taste perception, it means the emulsified in saliva, the food. Okay, then this process is a prerequisite for the sense of perception of taste. This is due to the presence of water and lipocalins in saliva. Speech, oral tissue moist and well lubricated facilitates speech, helps in vocalization and communication skills, helps in tissue repair. How? By the IgGs and all the factors. It helps in excretion also. Pancreas and gastric glands. The saliva is generally not lost to metabolism and it is reabsorbed in the GIT. Many substances from blood reach the saliva. Thus, saliva can be considered as a route of excretion. That's why. Okay, then let's go through the classification. It is very main classification. The salivary glands can be classified on the basis of function and histologic appearances into three types: serous, mucous, and mixed. Mixed has these serous and mucous acids both. On the basis of size, major and minor. On the basis of location, we can say labial, lingual, buccal, mandibular, if they are in mandible, etc., etc. Now we'll study about the serous salivary glands. These are glands that are predominantly made up of salivary acini, which are serious. Not serious, it's serous. Okay. The serous acinis are the glands or the cells which produces the saliva, different types of saliva. These are pyramidal shaped with a broad base and a basement membrane. The apex faces the lumen and they have a spherical nucleus placed at the basal region. The apical cytoplasm of these cells show accumulation of secretory granules. So this contains the secretory granules. The secretory granules are 1 micrometer in diameter with a distinct limiting membrane. These are embedded in the tissue section. Plastic embedization, stained with toluidine blue or specific cytochemical techniques. And these have some granules that are closely opposed to each other, but remain their individuality. These are known as zymogen granules and are formed by glycolytic proteins that are released into a vacuole. In electron microscope, the immature granules appear paler in density as compared to those electron dense granules that are maturing and moving towards the luminal plasma membrane. Okay. And then these SNL cells also contain small eosinophilic granules which appear more dense and concentrated towards the tip of the cell. These are the zymogen granules. Okay. Interglobular and intraglobular. Interiobular and intralobular ducts are also noticed. The parotid gland is a predominantly serous gland. So it produces a watery saliva. So serous gland produces the watery saliva. The only minor salivary gland that is serous in nature is the von Abner salivary glands. We'll study about it later. These are the types of the salivary glands. Okay. See the fibrous septum, the interlobular ducts, the interiolobular ducts. Okay. Intra and interlobular. These are the differences. And there is the salivary glands, serous salivary glands. This is not more visible because it's so confusing because of the HNE stains. But in this, you can clearly see the SNI, okay, the serous SNIs. In these granule type things, these granule type things are the zymogen granules. Fibrous septa here, this continues like this, and then it goes like this, and here, okay. These are interlobular ducts, and these are intralobular. Interlobular is in the main section, and intralobular is within the SNI. Okay. Now, the mucous salivary glands. Mucous type of salivary glands SNI are predominantly noticed in these glands. Mucous acinar cells are also pyramidal in shape. However, the nucleus is flat and pushed towards the bottom of the cell. Cytoplasm rarely takes up any stain and appears pale or empty on routine HNA stains. Interlobular and intralobular ducts are present. Sublingual salivary glands are predominantly mucous in nature. Okay, all the minor salivary glands except the von Ebner are mucous because the von Ebner is serous. As you can see in these are so confusing. Mucous SNIs, interlobular ducts, the fibrous septum here, all the same as the serous, but these differ in the zymogen granules. These does not have the zymogen granules. Okay. The mucous acinides, these don't have the zymogen granules, which are like these here in the serous one. This is the difference between these mucous and serous cells. Pyramidal or large, cuboidal in shape, flattened nucleus at base. Okay. But in this, pyramidal in shape with narrow apex. Here, the large flat cuboidal shape, but here the apex is near the lumen or the conical shape. Spherical nucleus near the basal one third, but flattened nucleus at the base. Apical portion of cell appears empty, contains mucous vacuoles, but here the apical portion contains zymogen granules, as we have seen. Apical portion stains weakly with HNA, but here strongly. Here apical portion stains strongly with carbohydrate stains like PAS and Alcian blue, but here shows numerous eosinophilic secretory granules that stain with toluidine blue. Apical cytoplasm not sealed, mucus can spill into lumen, apocrine secretion. Here, merocrine section, no loss of cytoplasm. Granules as a string of pearls. The mucus and serous acini, elliptical or tubular shape, larger in size, but here spherical in shape and smaller in size. Have a smaller stellate lumen because it's smaller in size. Lacks intercellular canaliculi. Intercellular canaliculi as presence. Secretion, no enzymatic activity, acid phosphatase, esterases, glucuronidases, etc. produces more carbohydrate components than proteins. Secretory glycoproteins have O-linked oligosaccharide chains, but these produces secretory proteins that are carbohydrates content less. Secretory glycoproteins have N-linked oligosaccharide chains. Here O-linked, here N-linked. These carbohydrate components that proteins, but here the content is less. In electron microscopy, prominent Golgi regions located between nucleus and secretory droplets, but here these are not like these. Extensive ARIA is present. Then there comes the mixed salivary glands. The mixed salivary glands contain both in serous and mucus. Few serous are seen arranged as a crescent-shaped structure on top of some mucus acini. These are called serous dimelunes, as we have seen in the previous images. Dimelunes of Genesis, yeah, Damihalt luminum. It is now known that dimelunes are artifacts that arise due to traditional tiny preparation methods. The tissue prepared using liquid nitrogen osmium tetroxide shows serous and mucus cells aligned normally with the sinus. In conventional tissue preparations, mucus cells swell and push the serous cell outside, resulting in dimelune appearance. So that's how dimelune appearance gets its shape. Submandibular salivary gland is a mixed salivary gland. Okay, this is the typical ones. If you can understand these, you are genius. These are the schematic representations. As you can see, there are some zymogen granules, and these are mucus. Okay, the mucus acini, the serous acini. These are the serous dimelunes. Here you can see short proportion of serous over the mucus acini with some zymogen granules, and all types of ducts are present here. Then the ductal system, it is very main thing as some point of view. And the short note can come on this, or a long note can come on this. Classify the salivary glands on the basis of histology functions. So you have to write all the three types. Then the ductal system. Okay. So salivary gland contains a system of ducts that acts as canals in which saliva flows from the secretory unit to open into the oral cavity. Some parts of the ductal system also modify the salivary composition. Intercalated ducts are usually found in an intralobular location, okay, within the lobule. But these are the smallest in the ductal system and carry secretions from the acini to the striated ducts. These are lined by a layer of low cuboidal cells. Then comes the striated ducts, form the major component of the ductal system and are mostly located intralobularly. These conduct saliva from the intercalated ducts to the excretory ducts and also play an important role in modifying salivary composition. So this one helps in modifying, not this one. These are lined by columnar cells with centrally placed nuclei. These are the schematic representations. These are intercalated. These are striated. The striations are present. So, and these are the excretory ducts. Okay. This is the HNA. As always, you can't understand this. See, these are serous dimelunes, mucus acini, serous dimelunes, serous acinas, intercalated ducts. The basal portion of these cells contains fine striations which give them their name. Electron microscopy reveals that these striations are deep infoldings of the cell membrane in the basal region. What does this mean? The deep infoldings of the cell membrane in the basal region. From the basal region, these are deeply folded inside or the inward with numerous mitochondria in between, because the salivary secretion is always taking place. Even when you are sleeping, you are talking, you are eating, you are not eating, you are dancing, or whatever you are doing, the secretion will always take place. It is continuated process. So these require many mitochondria. Excretory ducts are the terminal structures of the ductal system which ultimately open into the oral cavity. These are usually seen in the interlobular connective tissue septa, the fibrous septa. These are lined by the stratified cuboidal, stratified columnar, pseudostratified columnar, or stratified squamous epithelium. These all are in one. These ducts help pour the secretions into the oral cavity and also contribute to the salivary modification to a lesser extent. Okay. This is a large excretory duct you can see here. All the secretions come here from the nucleus acini and serous acini, all comes here. Okay. And then it all goes into the oral cavity. This is the difference between them. Location and size. You can always write these and you can use these lines as your headlines in your paragraphs if you are want to answer in the short notes. If short note comes on any one of these, if long note came, you have to write all these points. Okay. You can take it as an exam point of view. These are the differences between the parotid, submandibular, and sublingual. These three major salivary glands, and these are the differences in them. And the main difference is which type of cell, purely serous, mixed, or mixed, and what type of viscosity of saliva is there, the serous, the pseudomucus or the mucus, thick and viscoid, and all the intercollected, and which type of ducts are present in these, and with the ducts names, Stensons, Wartons, and Bartholins, and all. You have to remember all these. If the short note came, you have to write all of these things. The minor salivary glands, these are four in number, labial and buccal glands, present in cheek and the lips portion. Glossopalatine in the palate, palatine also in the palate, but glossopalatine behind the hard palate, the junction of the soft and hard palate. If you have studied posterior palatal seal, you can search it on Google. You will know about it, glossopalatine. And it has a very remarkable relativity with the third year. So you have to thoroughly study about it. Lingual glands, von Abner glands. The major and minor differences between them. You can write these. It won't come very commonly. It is uncommon to come in your exams. So you can just skip it. Age changes. Acinar cells show reduced activity which results in reduced salivary secretion, leading to xerostomia or dry mouth. It will lead to the dry mouth and it will cause irritations and soreness of mouth and all. Salivary secretion becomes thicker and contains fewer proteins and IgA. So in the old ages, you get more and more infections in your oral cavity. So oral hygiene must be good. Fibrosis and fatty infiltration are seen in the glands. SNR cells show oncotic changes. Oncotic means the cancerous changes. So thank you for listening to me for this long. I hope you found this video useful. If you want the PPT, follow the link below. And please keep checking our YouTube channel for more. We'll give soon the phases of decommission. Thank you so much.